Aloha, welcome everybody. It is my honor today to introduce Sean Kirkpatrick's mentor, Dr. Mark Hughes. Dr. Hughes is originally from Florida and received his undergraduate degree in biological sciences at Florida Atlantic University. Mark completed the graduate program at the University of Florida Gainesville and received his PhD studying laurel wilt disease. In 2016, Dr. Hughes moved to Hawaii to conduct his postdoctoral work with the Lisa Keith Lab on Rapidohea death. He currently works with the Pacific Cooperative Studies Unit on Rapidohea death and other forest pathogens. I now hand the mic over to Dr. Hughes.
Thank you, Lisa. Good morning, good afternoon, everybody. It's my pleasure to introduce Mr. Sean Kirkpatrick. Sean was born and raised in a rural farming community in the San Joaquin Valley of California, and upon graduation from high school, enlisted in the United States Marine Corps. His trip to the Marine Corps Recruit Depot in San Diego was his first flight in an airplane. And from that opportunity, the Marine Corps gave him an opportunity to travel, which was very important to him. And he received his orders for his next stationing in the Marine Corps Air Station at Kaneohe Bay in Hawaii. And from there, Sean has grown to love the unique place that is Hawaii. His exposure to different cultures that he received during his enlistment made this decision to remain in Hawaii a very simple one. Sean has committed himself to preserving the unique landscape of Hawaii. So Sean received his Bachelor's of Science from Hilo and is now at the final stages of his work in the TCBES program. Those are fingers crossed, not I'm sorry, with the fuzziness. And the TCBES program, and his studies are focused on post-border biosecurity with the goal to better preserve and maintain the health and resilience of our forest. Sean has started working with me in my lab in late 2021. So he's my first intern, so that's great. Where he learned and executes many of the plant pathology lab and greenhouse techniques that are routinely used in our field. His assistance has been invaluable in furthering our research on management strategies against Rapidohea death. And additionally, Sean has been instrumental to our investigations of an unknown and mysterious dieback of strawberry guava, where he's completing Koch's postulates right now and to help determine if fungal pathogens are involved, and if so, what are they? So with that, I'm very lucky to have Sean working in the lab, and he's been a great help. And so I can't say that enough. And with that, I will leave this to you, Sean, and good luck.
Thank you very much. So good afternoon, ladies and gentlemen. Thank you for being here today. My name is Sean Kirkpatrick, and I am a member of the 2019 cohort of the TCBES program. I'll be sharing with you my internship experiences on post-border biosecurity efforts in Hawaii. The host agency for my graduate internship with the United States Department of Agriculture Forest Service, located within the Pacific Southwest Research Center at the Institute of Pacific Islands Forestry, from here on referred to as IPIF. This agency works closely with several different organizations in support of conservation in Hawaii, such as the National Ecological Observatory Network, the Mauna Kea Forest Restoration Program, and the Hakalau National Wildlife Refuge. One of my mentors for this work is Kealoha Kenny, a research ecologist with IPIF. Kealoha is an alumnus of the University of Hawaii at Hilo, where he received a BA in Geography, GIS, Remote Sensing, and Biogeography. Dr. Kenny completed his graduate work at Brown University, where he received his PhD in Ecology and Evolutionary Biology. The research he currently conducts is important for advancing theoretical and applied understanding about the threats facing Hawaiian and Pacific Island ecosystems. The team's research efforts have resulted in new tools and strategies that can be used to protect, conserve, and sustain the natural resources and services that forest ecosystems provide. The other mentor with whom I worked closely is Mark Hughes, a researcher with the Pacific Cooperative Studies Unit at the University of Hawaii at Manoa. Dr. Hughes has worked on the subject of tree diseases in his doctoral work, specifically examining laurel wilt disease and the development of a natural resistance program. He has been able to apply his knowledge to supporting conservation efforts in Hawaii, working on different aspects of rapid ohia death disease. My previous professional experience prior to entering academia was in the military and law enforcement fields. When I moved to Hawaii Island, I became involved in agriculture, working with large livestock, and eventually created a business around beekeeping. When I started working as an apiarist, I was exposed to the negative impacts of dealing with invasive species, such as the West African small hive beetle and the Varroa mite. I increased my knowledge of integrated pest management strategies to mitigate these challenges. This activity fostered in me an interest in the environment and how to preserve the unique landscape of Hawaii. Unfortunately, I developed a sensitivity to bee stings, and I was allergic to bee stings. And if I continued working in the apiary, it most likely would have ended my life. So I went back to school. When I began my collegiate career, I started in the Tropical Forest Ecosystems and Agroforestry program at Hawaii Community College. This fantastic program solidified my desire to work in the conservation field here in Hawaii. I continued my academic journey, receiving a bachelor's degree and now a master's degree from the University of Hawaii at Hilo, always with an eye on working to preserve Hawaiian ecosystems for future generations. The focus of my graduate work has been biosecurity. So, biosecurity, it encompasses a spectrum of policies and actions that minimize risk from invasive species. It's critical in addressing the challenge of nuisance pests, plants, and pathogens. The Hawaii model, as denoted in the 2027 Hawaii Interagency Biosecurity Plan, takes a broad view of biosecurity, where it examines needs in pre-border risk mitigation, border interception, and post-border detection and response to protect the state's agriculture, environment, economy, and health. I had been exposed to the problems caused by rapid ohia death and the detrimental impact this disease was having on many Hawaiian native flora and fauna in an undergraduate internship with the Pipes program. Go Pipes. So I chose to focus on this issue. My work was looking at post-border biosecurity with fungi that cause tree disease. Project specifically was dealing with the invasive species responsible for rapid ohia death disease, otherwise referred to as ROD. The disease-causing pathogen are fungi in the Ceratocystis complex. The two species of fungi of interest are Ceratocystis leucoohea, which means ohia destroyer, which causes a systemic wilt, and Ceratocystis huliohea, which causes a localized canker. Ceratocystis is a fungal genus with several species that cause rot disease of agricultural crops and vascular wilt and canker stain of wooden plants. A typical vascular wilt pathogen moves through the xylem but does not invade xylem parenchyma or ray cells until the host metabolism is disrupted and the tissues surrounding xylem vessels die. This threat to the native forests of Hawaii was first positively identified in 2014 and has impacted more than 175,000 acres of forest that contain Metrosideros polymorpha, otherwise known as ohia lehua. This disease has the potential to cause massive damage to the ecosystem of Hawaii negatively, impacting the organisms that rely on ohia, as well as reducing the productivity of the watershed and human agricultural activity. And focus of my project was to examine the efficacy of current biosanitation procedures designed to limit the translocation risk of viable ceratocystis spores. To determine the effectiveness or the suggested biosanitation procedures, which includes washing the undercarriage of off-road vehicles for limiting spore translocation, I needed to sample vehicles that had visited areas with known positive ceratocystis presence. I planned to arrange with Forest Service personnel to collect samples from vehicles that had traveled off-road at known rod hotspots before and after the biosanitation methods were employed. First, I wanted to determine what is the abundance of viable spores in the environment. One of the abiotic methods of movement for the spores is on wind-borne frass, a waste material generated by wood-boring insects. To accomplish this, I plan to deploy and maintain an active environmental monitoring system which collected frass for evaluation for viable ceratocystis spores. Another avenue of research was to determine if anthropogenic activity was moving viable spores around the island. The ceratocystis complex of fungi are known to have a wide range of host plants, such as cacao, sweet potato, and members of the Syngonium family. Because of this wide host range of plants, I thought it would be important to determine if the prohibition on movement of ohia was effective at limiting the translocation of rod-causing fungi. On the island of Hawaii, the municipal landfill facilities located in Hilo have a green waste recycling processing facility that produces mulch for use by the community. I thought that there could be the possibility of other host plants being processed in the green waste facility, which could lead to translocation of viable ceratocystis spores. Spore viability testing method is called carrot baiting. It entails taking a common carrot, slicing the taproot into discs, placing the sample in question between two discs, creating a sandwich, and securing the sandwich with a parafilm. Carrot sandwiches are then placed in storage, labeled, and kept lidded at room temperature out of direct sunlight. Carrot baits are visually inspected every 3 to 7 days for the duration of the 30-day bait period. The carrots provide a semi-selective growth medium that aids in growing the specific cells sought. The selectivity of this media can be increased by soaking the carrot wafers in antibiotics such as tetracycline, ampicillin, and streptomycin prior to exposure to the sample. The primary reproductive structure that is used as a diagnostic tool to identify ceratocystis is the parathecia, a round or flask-shaped fruiting body with a pore through which the spores are discharged. A sample was a positive if parathecia formation was observed. Positive samples were then analyzed using quantitative PCR to definitively identify the species as either Leucohea or Huliohea. I began the TCBES program with high hopes for my internship. Then, in spring 2020, the world changed. Almost overnight, all non-essential activity was shut down. The stay-at-home order was put into effect, and in-person activities ground to a halt. Over the next several months, meetings moved to distance-based platforms such as conference calls and Zoom meetings. I participated in the monthly ROD working group meetings, which involved various groups and individuals working in ROD, both here in Hawaii and on the continent. This included the invasive species committees of each island, the Department of Land and Natural Resources, Department of Forestry and Wildlife, outreach and education personnel of the College of Tropical Agriculture and Human Resources, and the different scientific researchers in Hawaii and elsewhere, examining aspects of the disease. The group of outstanding people have been instrumental in generating support for ROD efforts in the state legislature through dedicated funding. This group has also been intricately involved in advancing Senate Bill 2059 and House Bill 2202, which calls for the designation of Ohia lehua as the official endemic tree of Hawaii. I also participated in the IPIF rod and ungulate fencing bi-monthly meetings to stay up to date with the various projects undertaken by IPIF, associated personnel such as the Flint Hughes Lab, who was examining the links between ungulate activity and rod movement and severity. Even though the restrictions associated with COVID severely limited my ability to conduct the planned activities, I was able to stay connected and informed about the efforts undertaken to address this disease. The work conducted on ROD involves many stakeholders, including public and private landowners. Earning and maintaining the goodwill of these varied stakeholders is an important factor in successful efforts. These stewards of the land often have a very close relationship with the lands that they interact with, providing an excellent source of observational data. Often, the first indication of the spread of ROD to new areas has been from these individuals. As a show of support and solidarity, the scientists of the Kenny Lab often participate in various community efforts. These images were taken as part of a Lo'i restoration undertaken in the Pololu area by Hawaiian Cultural Center of Hamakua. This site was very noteworthy in that the Lo'i were constructed in terraces along a steep hillside with large stone walls forming the banks and walkways of the Lo'i. Vascular wilt, wood stain, and stem cankers are the most characteristic symptoms of infection by Ceratocystis species in wooded trees. Ceratocystis species have telomorph, sexual, and anamorph, asexual stages of reproduction. The telomorph stage can be recognized through the presence of a small, dark through to light colored globose or spherical fruiting body known as ascocarps or ascomata, which are typically parathecial, which is the image to the left. The base of the ascocarp is enlarged and they have long necks with hyphae on the tip. This structure is the primary diagnostic indicator of the presence of viable Ceratocystis spores in the sample. The ascospores in the middle are hat-shaped mass of sticky spores that easily stick to the legs and bodies of insects that feed on these fungi, facilitating spore dispersal to other trees. Staining, or sap streaks, can start from where spores have invaded freshly damaged tissue. They rapidly germinate and colonize the xylem and phloem, absorbing nutrients from the xylem parenchyma. The discoloration is caused by a combination of host response chemicals and the pigmentation of the spores and hyphae of the Ceratocystis. Once present in the vascular cylinder, the hyphae of Ceratocystis species can then move systematically into the cambium and inner bark, killing these tissues, causing a canker. Disease spreads depends upon inoculum production and dispersal. The presence of susceptible hosts and suitable environmental conditions. Ceratocystis species produce several different kinds of spore inoculum and exploit a range of dispersal mechanisms, which may vary according to the type of spores produced. Like many filamentous fungi, the spores of Ceratocystis species infect their hosts through an infection court, often a wound, where the spores germinate after deposition. Wounding is critical for infection to occur. As the COVID restrictions dragged on, it became necessary to alter the focus of my internship to allow for the performance of the graduate requirements. At this point, the opportunity to work with Dr. Mark Hughes and his more general forest pathology focused efforts arose. Dr. Hughes was working on an aspect of rod disease knowledge generation, as well as other tree diseases. This aligned well with my general biosecurity goals. Through our discussions, we decided to focus on collaboration on three main areas: support of the Ohia Pruning Wound Susceptibility Project, the Strawberry Guava Dieback Project, and the Ohia Canker Disease Project. By participating in these projects, I was able to contribute to long-term results. The projects were at different stages of completion, which allowed me to be exposed to the preliminary, preliminary, midpoint, and culmination points of these efforts. A growth medium or culture medium is a solid, liquid, or semi-solid designed to support the growth of a population of microorganisms or cells via the process of cell proliferation. Different types of media are used for growing different types of cells. Potato dextrose agar, PDA, is used for the cultivation of fungi. PDA is composed of dehydrated protein infusion and dextrose that encourage luxuriant fungal growth. Agar is added as the solidifying agent. Culture media is of fundamental importance for most microbiological tests to obtain pure cultures, to grow and count microbial cells, and to cultivate and select microorganisms. Powdered media for agar cultures must be dissolved in distilled water and sterilized using an autoclave, where it undergoes heating under pressure for a specific time prior to pouring into sterile petri dishes using aseptic techniques. The sterile environment was necessary to avoid contamination of the culture. The most common source of contamination is bacterial or microbial animals in nature. Working with growth medium was a new experience for me that allowed me to expand my familiarity with using an autoclave and working in the antiseptic environment of the fume hood. Dr. Hughes has developed a system of color codes to identify the specific media present in the petri dish, as well as different amendments present in the media. I primarily worked with half-strength potato dextrose agar with various amendments. This formulation was optimal for controlling fungal growth rates and maintaining the viability of the cultures. When working specifically with Ceratocystis, a 10% V8 medium was used. The Ceratocystis really prefer the vegetable nutrients present in this type of media. The pruning wound project was designed to determine how long a wound to the plant was susceptible to infection by Ceratocystis. To accomplish this, saplings had a minor side branch of the stem cut with pruning clippers to provide a wounding site for infection. All plants for the trial were cut on the same date, then were inoculated with Ceratocystis leucohea materials at days 0, 3, 7, 14, and 28. When the plants had been exposed to inoculum for a predetermined period, the plants were visually evaluated for wilt, and the main stem was cut for analysis. The outer bark of the stem was removed within 20 centimeters of the inoculation site. The infection by the fungal pathogen was indicated by staining of the woody material. The length of the staining was measured to quantify the severity of the infection. Small slivers of the woody stem, at least 2 centimeters from the inoculation site, were collected and applied to carrot baits to evaluate for parathecia growth. A sample of the woody material was collected if the carrot bait was positive with fungal reproductive structures for qPCR analysis to positively identify that the disease-causing agent that was used for inoculation was indeed present in the sample. A real-time polymerase chain reaction is a laboratory technique of molecular biology based on the polymerase chain reaction. It monitors the amplification of a targeted DNA molecule during the PCR, not at its end, as in conventional PCR. Real-time PCR can be used quantitatively and semi-quantitatively. This method of PCR operates on the idea that a known organism's genetic material is present in the sample. There are fluorescing probes present in the PCR primers which bind to sites on the genetic material of the target organism of interest. This allows for the qPCR machine to detect a species, and the intensity of the fluorescence indicates the density of the DNA amplified. For these analyses, there's a positive and a negative control present. The positive control is Ohia genetic material. This control lets us know that the amplification did take place. In plot A, we see that the 2 target species Ceratocystis and Ohia were amplified. In plot B, ohia and lukuohia are present. In plot C, ohia and huliohia are present. And in plot D, only ohia is present. This slide shows what a negative result would look like. This could be an indication that the amplification did not or failed to take place. Land managers on Maui had observed a landscape scale dieback of strawberry guava, the cause of which was unknown. This was notable because the literature contains almost no known pathogens of Psidium catalanum. You may think that is a good thing because strawberry guava is an invasive species. This tree is in the same family as ohia, and as I learned, the host range of pathogens can be vast and can easily expand within tree families. In December 2021, Dr. Hughes traveled to Maui and collected samples of trees exhibiting the dieback. These trees had external lesions indicating possible infection by a pathogen. The samples were returned to IPIF for processing and culturing. Over the next few months, the endophytic fungi present in the samples were grown in culture. The first step was to take small slivers of the woody material from the samples and disinfect the external surfaces to avoid contamination. The slivers of the sample were placed within the media to promote growth. The fungi that grew were selected based upon morphology. As we can see from this slide, the variability in fungal morphology can be great. Once a fungal culture containing a single species of organism, once a fungal morphogroup was selected, I then set out to grow a pure culture. In microbiology, a laboratory culture containing a single species of organism. The next step was to culture a single spore. This is analogous to isolating a single individual from a population. This is done to aid in the genetic analysis and identification of the fungal species. DNA purification helps to extract genomic or plasmid DNA in the sample for analysis. Purifying DNA samples from contaminants reduces the probability of error when it comes to research results. The sample is homogenized to increase the success probability of DNA extraction. The primers used for extraction were targeted to the internal transcribed spacer region, which I will discuss shortly. The amplification of the extracted genetic material is accomplished through the use of primers to target specific gene regions of the DNA. The process takes place in a thermocycler. The temperature is carefully controlled to promote the amplification of the strands of DNA. Gel electrophoresis is used to ensure that the amplification of genetic material was successful. The procedure uses an electrical charge to promote movement of the DNA strands through the gel. Larger strands of DNA move more slowly through the gel. A DNA ladder is a solution of DNA molecules of different lengths used in agarose gel electrophoresis. It is applied as a reference to estimate the size of an unknown DNA molecule that was separated based on their mobility in an electrical field through the gel. The size of the molecule desired is 1,500 base pairs long. In the lower right corner, that's an example of the fluoresced gel. The top band shows a very tight concentration of genetic material, which shows that the extraction and amplification was a success. The bottom band shows several streaks, which can indicate that the amplification process might not have completed successfully. The internal transcribed spacer region sequence tells scientists the kind of genetic information that is carried in a particular DNA segment. For example, scientists can use sequence information to determine which stretches of DNA contain genes and which stretches carry regulatory instructions, turning genes on and off. The ITS region is the most widely sequenced DNA region in molecular ecology of fungi and has been recommended as the universal fungal barcode sequence. It has typically been most useful for molecular systematics of the species to genus level, and even within species, such as populations. Among the regions of the ribosomal cistron, the internal transcribed spacer, ITS, region has the highest probability of successful identification for the broadest range of fungi, with the most clearly defined barcode gap between inter and intraspecific variation. The sequencing of samples was performed by Eurofin Genomics LLC, located in Louisville, Kentucky. The current slide is an example of a genetic sequence of the ITS region. Once the sequence is returned from the lab, we need to format and manipulate the data for use in identification of the sample. The first step is to select the appropriate file type for use in MEGA. Next step is to clean up the data to increase the strength of the identification results. Usually at the beginning and the ending of an amplification run, the enzymes are not working at full capacity, so there will often be erroneous information encoded in the read. Since DNA is double-stranded, we need to apply these manipulations to both strands of DNA. Next, we need to align the two separate strands so that the appropriate sequence of base pairs nucleotides match, i.e. adenine and thymine, or guanine and cytosine. BLAST is an acronym for Basic Local Alignment Search Tool and refers to a suite of programs used to generate alignments between a nucleotide or protein sequence, referred to as a query, and nucleotide or protein sequences within a database. This allows for researchers to contribute to a growing database of verified genetic sequences. We enter the manipulated genetic sequence into the query box, and voila! End up with a list of possible matches. In this case, there is a 100% match for the ITS gene sequence. If there were inconclusive results for the ITS gene, we would sequence another genetic region for positive identification. Next is reviewing the literature and determine if pathogenicity has been established for the identified species. Of the 26 fungal isolates that were derived from the Maui samples, 21 were successfully sequenced and 9 species were identified. The most common fungi present in the subset were Pistolithiopsis species and Neophusococcum species. These 5 species were chosen for initial inoculation trials. Koch's postulates are 4 criteria designed to establish a causative relationship between a microbe and a disease. The postulates were formulated by Robert Koch in 1890. The 4 criteria are: 1, the microorganism must be found in diseased but not healthy individuals. 2, the microorganism must be cultured from the diseased individual. 3, inoculation of a healthy individual with the cultured microorganism must be recapitulated and capture the disease. And finally, 4, the microorganism must be re-isolated from the inoculated diseased individual and matched to the original microorganisms. Koch's postulates have been critically important in establishing the criteria whereby the scientific community agrees that a microorganism causes a disease. 5 healthy saplings of strawberry guava were inoculated with a plug of culture from each of the 5 fungi of interest. They will be observed long-term to see if the symptoms of the disease appear. The third project that I worked on was an ohia canker disease. Recently, there have been reports of a new disease impacting ohia that has similar symptoms to rod, but has a distinctive canker associated with it. The disease causes the formation of a canker that causes the infected stem to swell to the point that the outer bark tissue ruptures. Depending on the location of the canker, this may cause tree mortality. One of the trees that we were using for our pruning wound viability study was also exhibiting this canker wilt, or this canker disease. And when we went to grow out the different fungi present within the sample, in the upper right corner, we were able to also culture out very virulent ceratocystis spores. Often it's suggested that to maintain the virulence of fungal species, that the culture be processed through a host plant and recollected. I employed many of the techniques that I applied to the strawberry guava dieback project to this ohia canker disease project, such as sample processing, developing a pure culture, and single spore isolations. The work on this project is almost at the genetic extraction, amplification, and sequencing stage. By interacting with the multiple stakeholders of this project, my academic goals of gaining experience in real-world conservation management has been served. I have been an integral part of a team of researchers working on the front lines of biosecurity in Hawaii. Some of the deliverables that I added to my included creating a physical archive of 60 plus fungal isolates for genetic identification and use in inoculation trials, and creating a photographic archive of 60 plus fungal isolates for use with identification of unidentified fungal morphology. I would like to acknowledge the many outstanding people who provided their expertise and support in the completion of my professional internship. I would like to thank my mentors, Mark Hughes and Kealoha Kinney. Your patience, professionalism, and openness to share your vast knowledge with me was greatly appreciated. I would like to thank Sarah Knox and Grace Trednick for your welcoming attitude and making me feel like an integral part of the lab team. I want to thank Eva Brill from the Pacific Basin Agricultural Research Center for her technical assistance with the genetic protocols and equipment necessary for sequencing. And to the members of the ROD Working Group and ROD and Ungulate Fencing Groups, I appreciate all of the work that you do to preserve Hawaii's forests. And last but not least, I would like to thank the TCBES 2019 cohort for all the official and unofficial assistance you've provided in my academic journey. Thank you very much. Can I hear you, Lisa?
Once again, I'm 0 for 3 for this afternoon. Anyway, thank you, Sean, for your presentation. That was fantastic. And there's a lot of information in there. I would like to open up the floor for questions. I think we have time for maybe 1, maybe 2 questions. So if you have a question for Sean, please unmute yourself. Okay, I'm gonna be the first one then. So Sean, you've had this quite a journey from your work that you did before you went to school, through the forest team, through the geography department, TCBS, and you've experienced field work, greenhouse work, lab work. Out of all those experiences, what's resonated with you the most? Like,
well, I really did enjoy the I guess the lab work was really enjoyable. It was very interesting. It wasn't what I had anticipated when I was imagining what lab work would be like, but it was definitely a growth experience. But I like being out in the field. I like to, hear the birds, feel the wind, feel the rain.
Okay, all right, thanks for that. Does anybody have a question that you'd like to ask Sean?
I don't have a question, but I just wanted to say great job, Sean. That was a really nice presentation, and I'm really proud of what you've done. So congrats.
Thank you.
And so, what's the next step in the work that you've been helping out with?
Well, the next step would be preparing the ohia canker disease cultures for extraction, amplification, and sequencing, and then selecting inoculation sources.
All right. Okay. And are you going to be continuing on with this as a volunteer, or what's your next step in your journey?
That has yet to be determined.
Okay. Awesome. And if you, I'm sorry, I'm asking you all these questions, but I have one more. If you had any advice to give someone that was looking to get into the field of conservation, given your vast experience, what would you recommend?
Well, don't be faint of heart. Unfortunately, conservation involves a lot of not good news, and it can be very disconcerting to see that invasive species and pathogens are such a problem to our native ecosystems. But if you go in realizing that the work you do, you may not see the fruition of, but knowing that you conceivably have helped to preserve part of the landscape for future generations, that's definitely rewarding.
All right. Well, thank you so much, Sean, for your presentation and your words of wisdom. We are now going to take a quick 3-minute break, and I will see you all at 2:15 for our final presentation of the day. So I'll see you in a few minutes.